So, hello everyone. Thanks for coming. We are very happy to have with us Dr. Benos, who is an authority on computational biology, to give today's lecture. Dr. Benos comes with a very interesting background. He started from mathematics, where he received his first degree from the University of Crete in Greece. Then he proceeded to get his PhD in molecular biology, making a 180-degree turn, so to speak, from the same university. And then he went to the EBI in Cambridge, where and also in Moscow, St. Louis, where he got postdoc training in computational biology. And he's currently Associate Professor of Computational and Systems Biology with the University of Pittsburgh School of Medicine. And he's also the co-director of the CMU and University of Pittsburgh Program in Computational Biology, and the co-director of the Center for Translational Bioinformatics with the Departments of Computational Systems Biology and the Department of Biomedical Informatics at the University of Pittsburgh. His talk today is about microRNAs and the spectrum from disease markers and regulatory networks to therapeutic agents. I'm sure it's going to be a very exciting talk. It's a real pleasure to have you, so thank you for coming.
Thank you very much.
Thank you very much for the invitation. It's a pleasure to be here. I don't get to New York very often, but whenever I come, it's really good. I have shortened the Panagiotis to Takis because it's easier to pronounce. Don't ask me how this comes, but that's how it is now. So the talk will be about microRNAs today. microRNA is a relatively new subject in my life in the lab. We started working with microRNAs in 2007. And I will, during the talk, I will go through various algorithms as well as biological findings that we had over the years working with microRNAs. So the ultimate goal of the lab is not actually developing algorithms. This is a kind of byproduct. We are not computer scientists, we are more biologists, computational biologists. So our main goal is to understand the molecular mechanisms that are implicated in clinical phenotypes. We are in the School of Medicine. Everything we do has to have some kind of medical or clinical component. Our work involves development of computational algorithms, but as I said, this is the byproduct. Whenever we cannot solve the problem or answer the question with the existing algorithms, then we go to developing new algorithms. The central theme of the lab is related to gene expression, from transcription factor binding to microRNA and gene expression to more recently genetic modifications. And all that, of course, we use to make these pretty and, for the most part, useless curveballs, as you know, through the data integration. So we know very well that the results we find are very many, but then finding through these statistically significant results to cut down to the biologically relevant ones, it's not always straightforward. So the overview of the talk, I'm going to talk a little bit about, for those that they don't know what microRNAs are, if there are such people nowadays among us. I'm going to talk a little bit what microRNAs are and why we're interested in them. Then I'm going to talk about some algorithms we had on predicting the microRNA genes, and also a more recent algorithm of predicting microRNA targets. And then how we can incorporate gene and microRNA expression into gene networks, and how all this can be used to help us improve care for different disease patients or do biomarker development. Now, during my talk, when I say we did that, actually I didn't do anything. kind of royal we. So this is the group that did the whole thing. And actually today I'm going to talk the work of Harry Athanasiou, Claudia Coronello, who was a postdoc in my lab until a week or two ago when she returned to her home in Italy, in the research, medical research institute there. Grace Wang, who is now the oldest student in my group. Sabah Kadri, who graduated recently and went to the Broad Institute for a postdoc, and David Corcoran, who was my first student, and after he did the postdoc with Uwe Oller at Duke University, he's now the director of the core facility for data analysis there. So, by the way, if you have any questions, you feel free to ask me anytime during the talk. Some people think that they are afraid to do that because they are afraid that I'm going to lose my train of thought. I don't have any, so please feel free to interrupt me if you have any question you don't understand something. So what are microRNAs and why you were interested in them? So a couple of things we know about microRNAs. They are 60 to 80 nucleotides long in the pre-microRNA form. Then the functional form is 20 to 24 nucleotides long. These are single-strand, of course, RNA, non-coding RNAs. They can be located either in introns of genes or in the intergenic regions. They are mostly transcribed by Pol2, polymerase 2. Some of them are polymerase 3, but these are exceptions. And the role is post-transcriptional regulation of mRNA genes. A little bit for the biogenesis. They usually generated as a polycistronic, you would call them, transcripts, which they contain many of these stem loops. And the stem loops may contain a microRNA when they are processed further. These, some people believe they are polyadenylated, but if they are, this is a short-lived message, because even in the nucleus, the stem loops are cut off, they're transported in the cytoplasm, the loop is cut off, and then you end up with a double-stranded RNA, and then other proteins pull these two apart, load one of the two strands, or sometimes both strands, into the argonne protein, and then this takes the microRNA to find its target on the mRNA by virtue of base complementarity. Most of the people will look on the 3' UTR of these mRNAs because this is where we find the most targets, although targets have been found also in 5' UTR in off-breeding frames and so on. Now, why we're interested in microRNAs? Although some very bright people do not believe in microRNAs, like Larry Goff, which is my spiritual grandfather in a sense. He was the PhD advisor of my postdoc advisor. Where he famously said that cells do not seem to have learned the central dogma, DNA makes RNA makes protein. He was trying to convince that RNA is all useless, we should abandon working with mRNA and working only with the proteins. He had a point there, and that's a long discussion, but he's developing aptamers that act for recognizing proteins. And he showed that with the proteins you can have a pretty good diagnostic test, and he's very feasible doing that. He has a company doing that. Anyway, so many smart people believe that most of the microRNAs do not do anything, and there are few of the microRNAs that are out there that they do something, and we have proven them, and we falsely generalize to all of them. Despite that, there is an increasing number of papers that show, like in 2010 we had 542 studies that implicate specific microRNA genes to diseases, and there was a database generated on that. And don't forget that it's not that long ago, maybe it is long ago now, that all this non-coding part of the genome was considered as junk DNA from some people. And when Sidney Brenner was asked about the junk DNA on the talk he gave in Washington University in 2002, he said that there is a difference between junk and garbage. Junk is what I keep in my basement in the hope that one day will become useful again, until natural selection, in his case it was his wife, goes there and turns that into a garbage. So you can view microRNAs in the similar light. Okay, so I hope I convinced you that microRNAs are important to study, and that is what drawn us to that. We started actually with a project, a collaborative project with a faculty at CMU, Veronica Hillman, who was interested in studying, we were interested in studying the implication of microRNAs in the development of sea urchin. Sea urchin, the problem with sea urchin was that the genome was not sequenced back then in 2007, so we didn't know where to look for microRNAs, and there was nothing nearby sequenced that can guide us through evolutionary conservation to find the microRNA genes. So we had to have a method that does not depend on evolutionary conservation to be able to look on microRNAs and on parts of the genome and say whether they code for a microRNA or not. Doing deep sequencing was not there, was not an option until a few years after because of the cost. And clip methods were non-existent, so there was no other way to look for microRNAs. So in that case, what we looked, as I said, this is the stem loops of when they are produced in the genome. Most of the time, or not most, but many cases, you get multiple of those. But we were interested not on the whole thing, but to classify whether a particular stem loop might contain a microRNA or not. So we were aided by having a number of microRNAs in the database at that point, and then when we stare on this stem loop long enough, it occurred to us that actually the stem loop can be divided in 4 different parts very conveniently. It's the loop, it's the microRNA, assuming we know where the microRNA is for the training data set. Then it's the region between the microRNA and the loop, and it's the region after that. Now, each of those, as you can observe, they have a number of matches, they have mismatches, and they have insertions or deletions they can have, each of them. And we expect these to occur with different frequencies on different regions. Furthermore, one might expect that the number of, let's say, CG base pairs might be different in this region than in this region, right? So in principle, every one of these regions can be modeled with what the computer scientists like to call a hidden Markov model. So, which is nothing more than some probability calculation. So now, let's say on the microRNA, you can have match, mismatch, or insertion deletion, and each of those will generate a number of base pairs, let's say, right? From that. And this can be learned from some training data set, which is the known microRNA that we had at the time. Now, because we have 4 hidden Markov models in a hierarchical way, you can start with the loop, then you have to proceed to the extension, you have to proceed to the microRNA, you have to proceed on the primary extension, and each of those will have a stage of match, mismatch, and so on with different probabilities. This is called the hierarchical hidden Markov model. Okay? So we developed the hierarchical hidden Markov model. It's about it. We used positive examples, the human microRNAs in the microRNA registry at the time. Negative examples, we folded hairpins derived from coding regions because, as I said, it's less frequent to find microRNAs in the coding regions. And our alphabet was, as you can imagine, AU, GC for base pairing, and GU as well. Maybe it's not very well known to the non-RNA people, but G and U can base pair in the RNA world. Different energies, but they can base pair. And we had the mismatches and the insertion deletions. We tried the 2 well-known algorithms for training that, Baumwelz and maximum likelihood. Baumwelz did a little bit better in terms of arrow sequence. Everybody knows what this curve is? Yeah? Who doesn't know? Okay, so here we kind of look on the specificity. So you can imagine that as how many of our predictions, so with sensitivities, how many of the correct predictions are, how many of the predictions are correct. Specificity is, sensitivity is true positive, it's how many of my correct, what is the percentage of the correct predictions over all the positive examples. This one is how many of my, what percentage of all my predictions are correct. So the one says how many of the true things that are out there I can predict. The other one says how many of those I predict are true. So these usually are opposite, and depending on the threshold that you put, you get some lines, usually like that, hopefully like that. If you didn't have any way to predict, and it was a completely random choice, the line would be from 0, 1 to 0. The perfect classifier will go up to 1 immediately, and then will go straight. So between the classifier that goes up to 1 immediately and then straight, and the random one, which will be the line from here to there, then anything above that is good, anything below that is really bad. Okay. So, and most of the times you measure the success by looking on the what's called area under the curve. So what is the area that you have here? As you see, the black line probably has a little bit more area than the red line, but they are pretty much the same. So when we applied it on different species, we found that overall it performs very well, and we compared it with the support vector machine, I think, that looked on prime nucleotide frequencies, and it was better at the time. So this was published on the proceedings for the Asian Pacific Conference in Biocomputing. And the summary of that part is that the probabilistic modeling of microRNA precursors can be used to efficiently classify microRNA containing stem loops, even when you go in long evolutionary distance. So, which was our objective. Okay. Any questions on that? Okay, so now let's move from the prediction of the genes to the prediction of the targets. That's a more recent work, still unpublished. We hope it will be published soon. So the things you need to know about the targeting is that this is the microRNA on the bottom. So the first 6 or 7 7 or 8 bases, like up to here, the targeting is supposed to tolerate very little number of mismatches, maximum 1 it says here. Although on a recent biophysics study by Isidori Goutsos and his colleagues, so that you can still modify the bases on the what's called seed region. This is the seed region. And you still get a very stable complex. They did that by molecular dynamics simulation. But anyway, that was the current theory. Target scan focuses mostly, it's one of the target prediction programs, the most efficient so far, although some other people might disagree. But focuses only or almost exclusively on the seed region. Then there is the second part of the microRNA, which there it can tolerate more mutations. Okay. So that's the general idea. There are many algorithms to predict microRNA targets. Here is just a sample. Pita and Miranda. Miranda is the algorithm developed by Chris Anders' group and looks on the actual thermodynamics of the binding to decide whether a microRNA might target an mRNA. Okay. It's a one-to-one. It's like these two things are on a cube and they are allowed to find each other and then it calculates some kind of energy. Okay. That's the premise of Miranda. Pita is similar, only it says, look, if the target is embedded on a stem of a stem loop, then the true binding energy will not be the microRNA to the target, but it will be the microRNA to the target minus the energy I need to pull the two stems, the two parts of the stem apart. Okay, makes sense. It's not very efficient algorithm from what I have read. Target scan, as I say, it uses thermodynamics based on the duplex prediction, but focuses on the seed region. And MIRES-ER is a more recent algorithm by Christina Leslie in Sloan Kettering, which uses a support vector regression to model the microRNA targeting. So there are many other algorithms that they do predict, and they all have a common characteristic. They don't overlap very much in their targets. So their targets, as you can see, these are two other studies, 3 algorithms, and you get less than 1/5 of the targets overlapping. And if you add more, then it goes even less. Okay. So that is a big problem. Yes, sir.
What about overlapping with the true gold standards?
There is no true gold standard.
But are there for some selected genes, people know whether there are some targets?
There are very few. The problem with the gold standard of the microRNA is that there are very few verified targets, and these are verified. So in order to truly verify the target, you need to find the target, then mutate the target of the microRNA and put it on a expression vector and measure differences in expression. This is a very laborious experiment. There are not too many out there for training of all the 1,000 microRNAs that we have. Right? Most of the people, what they will do, they will overexpress a microRNA, they will knock down a microRNA, they will see what other genes are changing. But this might be direct target, might be indirect target. It's not exactly clear. And also when you overexpress, there are other problems, like you oversaturate the argonaute protein, so then the other microRNAs that they are not expressed that highly might lose their targets completely because they cannot associate with the argonaute protein anymore. There was a work by Chris Ander, I think a couple of years ago, that solved that. So this is a problem. This is one of the problems on finding the target. There are some of them, they are confirmed, but on different cell types, and as I will show, the microRNA concentration, the microRNA expression does play a role, and I will show it in the next slide. So it's very difficult to say what are the targets. All of them, to some extent, they saw when they published some success on some gold standard that they have put out, right? But there is no standardized yet. Okay. So one of the reasons I believe that the overlap is not great is they don't consider, for the most part, these algorithms, the microRNA expression as part of the binding model. So what I mean by that, a gene might have 5 potential targets of let's say microRNA near 10. Okay. Each of these targets will come with its own binding affinity to this microRNA. Not all the same. When microRNA 10 is expressed at low levels, perhaps only one of these targets will be bound, the one with the highest affinity. And as this microRNA expression increases, then suboptimal targets in terms of thermodynamics will start get bound because the main target is already bound, and it's what's called target exclusivity. Once you have a target bound, you get more of these microRNAs, they cannot go and bind at the same thing because this is already defined. So you have this target exclusivity. It has to go to all the other suboptimal targets and you saturate it that way. So also another thing is that although we do tend to believe that this is the expression level of the gene that is targeted, microRNA starts expressed, this goes below detection levels, and this might happen as well, but there are other scenarios where the microRNAs are useful for the cell. So for example, here you have this from Andrea Califano paper. So you can imagine that the cell has 2 levels for mRNA. One is the low level, beyond with the mRNA is considered silenced. And then there is another level where if you go above that level, then the protein will become deleterious for the cells. So the microRNA might be used just to break the level from deleterious levels back to the normal levels. So the regulation here might not be so severe. Okay. And it is noted that each microRNA does not really shut down a gene except in few cases, but most of the time the effect is more moderate. This is also, sorry, this was not the Califano, this is the Califano paper, this was the Partel paper. So there is also what's called response modulation, where you can imagine you have 2 genes here, they both have targets for different microRNAs. What happens if this gene goes out of the game? All the microRNAs that were occupied by this gene will now be free to go to different targets. So will more downregulate this gene, or they might hit other genes. So taking one gene out of the cell might have indirect effects on the microRNA regulation of other genes. Okay. So the bottom line here is that everything in the cells happens quantitatively, not binary. It's not that microRNA X regulates gene A. MicroRNA X regulates gene A if the concentration of X goes above a certain level, otherwise it doesn't regulate. That's the bottom line, what I want you to keep from that. Okay, so one question that we wanted to address very early was, okay, if we try to answer the question, does mRNA NF-kappa regulated by this set of microRNAs that come out of the biological expression? Okay, that's a more realistic question that the biologists usually usually ask. They come and they say, I have these 5, 10 microRNAs, they are differentially expressed. Can you tell me which genes all of them regulate together? So now you go to one mRNA, and you might see out of these 10, you might find predicted targets for 5. Okay. How do you combine them? These 5 might be only barely above the limit to be called targets, but they are 5. So will they regulate the gene or not? Well, the answer depends on the concentration of these microRNA. So the first thing is, okay, when we try to combine scores for different microRNAs, one thing we need to know is, are these microRNAs additively or not? Are there any higher order effects? So our collaborators did some experiment where they overexpressed LET7B, they overexpressed MIR30B, and they overexpressed both of them. And we found out that actually only 132 genes, they seem to have these non-linear effects, at least statistically speaking. So for the most part, we can say that yes, the microRNA targets are additively. So can we improve now the modeling by taking the microRNA expression into the game? For that, we used a Drosophila Argonaut immunoprecipitated data set. And the reason that we chose that, it was because it was exactly what we needed for our project. So what Victor Ambros and his colleagues did was the following. They took the Argonaut protein, cross-linked the mRNAs and the microRNAs to the Argonaut protein, and they immunoprecipitated the whole complex. Right? That was before the clip methods. So they precipitated everything. They released the mRNAs and the microRNAs from the Argonaut protein. They separated high molecular weight, low molecular weight mRNAs, microRNAs. For the mRNAs, they throw them on the array, and they measured how many copies of each mRNA was associated with the Argonaut protein. For the microRNAs, they did deep sequencing. Okay, so you have now a set of microRNAs that targets a set of mRNAs, but you don't know which is which goes with which. Okay, but this is all that we needed. We needed a set, and we wanted to be able to predict whether they will regulate these guys on the other side. Now, these sets, the IP sets, usually are quite noisy. So what they did an extra experiment for us. They did it for us. It was good that they did. So the experiment was that they knocked out the Argonaut protein, and they looked which genes go up, which are the most potential targets, right? If you knock down the Argonaut protein, then all the microRNA regulation gets out of the equation. So those genes that you will see go up will be likely targets of these microRNAs that were the most important, right? So now we end up with 4 data sets. We have the data set of the mRNAs that they were bound to the Argonaut protein, and they go up after Argonaut depletion. That's our positive data set, right? Most likely true targets. Then you have the microRNAs that they were not bound to the Argonaut protein, and they don't change their expression after Argonaut depletion. These are the safest bets for the negative data set. And then you get other sets. You get this set, which is probably a mixture of direct targets and indirect targets. So these were not bound by the Argonaut protein, but they go up after the Argonaut depletion. So it might have been that they were bound to the Argonaut protein, but in very small amounts, that they were not detectable, or it might mean that they were indirect targets. Okay? And we have also the alternative. So anyway, so this was our main data set. Most of the data that I'll show you, it was based on this data set. For testing purposes, we also used the PAR-CLIP data set. Now the PAR-CLIP, so this was a Drosophila data set. This is a human data. The PAR-CLIP, they do something similar. They do again the cross-linking as before. But before they release the mRNA and the microRNA from the Argonaut protein, they clip them. They treat them with RNAs. So the thing that comes out of the experiment is the sequencing. Then they do deep sequencing. So it's the sequencing of this part of the mRNA, because everything else has been eaten out by the RNAs. Okay? So that's the data sets that we used. So, okay, how do you, first of all, the question is, can we improve the performance of beta and miRNA? Remember, these are the algorithms that they use thermodynamics to determine the target. but thermodynamics based on one-on-one, one microRNA, one target, like they were the only things in the set, in the cube. So this is the overlap on the Drosophila dataset of these two algorithms. When you combine all the targets together, so now you have all the, how many microRNAs we had? So we had, 38 microRNAs, 7,000 mRNAs. So these 38 microRNAs, we look for every gene. The naive method for combination of the targets is that, okay, this mRNA will be the target of these 38 if it is a target of at least one of them. Okay? Okay, so this is the overlap that you see between the two predictions. So almost 40%. Now, if you use the Fermi-Dirac equation, which takes into consideration the microRNA concentration when it determines the targets of the multiple things, as we did it here, this is the number of microRNAs, this is the number of targets of these microRNAs to a given gene, then as you see, the overlap increases. Right? And if you play a little bit with the thresholds and you look for ROC, this is how the algorithm performed in the naive kind of combination of targets. This is how the algorithm performed with the Fermi-Dirac combination of targets. Okay, so you see any progress? Yeah.
What response did you use? What was
what do you mean here?
What were the positive and negative?
So again, the positive and negative is the data set that I showed you before. So the positive is this data set, negative is this data set. Okay? On determining whether it's a target. Okay? So that's there is no algorithm development, just an observation that naive combination of targets is not as good as the Fermi-Dirac combination of targets. Okay. Okay. We saw that the same happens to a lesser degree if you apply the same principle on target scan and miR-SVR. Only now they don't use thermodynamic models, so that now the weighting is simple with the expression level of the microRNA. So the improvement is not as great as with the Pika and Miranda, but it's still, you can see some improvement. So now you get 4 tools where by including weighting in some way the expression level of the microRNA in determining the target of the cell, you improve their efficiency. Okay, so now you have 4 tools with improved efficiency. Why don't you combine them with the popular method of Alexander, which is the SVM? So the algorithm, that's the algorithm combiner, right? So the way that it works, you get a list of microRNAs and their expression levels. You can run Miranda, Pita, target scan, and miR-SVR with the improved models that I showed you before. You get some predictions, and then you funnel these predictions through an SVM, trained on the positive and negative data set that I showed you before. And do a target gene ranking. Okay, so let's see now how this combiner algorithm does in different data sets. So this, don't pay attention to that. This is a test on the same training set. We did also leave one out, cross-validation, it looks similar. This one is on the set 3 as positive examples, the ones that I told you that might be direct or indirect targets, right? So these were not used for training. So it's a kind of Drosophila, same species, but outside. And also as a negative data set, we used, on the training, we used only 142 examples out of the 4,000, and here we used the rest. Okay. So this is same species, but in a sense, it's an independent data set. So in the sense that the mRNAs, at least that they were used for training, they were not used, these are different. The testing are different than the training. This one is not a proper ROC because we didn't have negatives. It's argonaute immunoprecipitation on C. elegans, and we only have positives, so this you saw always an improvement. The only true thing that we can talk about is the human par clip data, where we have positive and negative results as well. And as you can see, especially in the area of high specificity down here, combiner does a lot better on that. The target scan, as you see, is very competitive, but unfortunately the target scan gives only a yes or no answer. The only way that you can get a score out of target scan is if you assess the evolutionary conservation. We didn't assess the evolutionary conservation here because none other algorithm uses evolutionary conservation. Okay. So actually for the human, they give you some context score. We did it, I have the slide if you're interested, I can show it to you at the end. Comer is still better, and it's statistically significantly better. So, okay, so the next thing is which of the components of this SVM really make Comer so better than the other methods. Okay. So here we did an SVM, if you like, evaluation of all the method combinations, right? For both the naive model, which is the red one, and the Comer model for combining the targets. This is method combination, like Miranda, Pita and Miranda together, Miranda target scan, and so on and so forth. So a couple of, and these are for the 3 data sets that we do have positive and negative example. So a couple of things coming out of that. One is that the Comer combination of score is always better than the naive combination. You see that the black dots are always better, higher performance than the red dots. Although the difference might not be very great in some of the combinations. The other is that, as I said, the magnitude of improvement is larger for the thermodynamics-based algorithms, like Pita and Miranda. Here is the Pita. These are, by the way, they are ordered by the difference in performance. Okay. So Pita and Miranda are the ones that they are benefited the most, and so on. Right? Here is the least benefited is when you have all the 4 combinations, all the 4 tools. Then the third one that you can see is that which combination will perform better depends on the data set, but usually the PMTS, the combination of all 4, is near the top. So PMTS here, the combination is, the efficiency is here, the area under the curve is here. It's almost the third one on the top. And PMTS here is probably the top one, the top scoring one, and PMTS here is, that one is also the second one, or the first one. So these are the conclusions for the Comer and the Comer performance. So, do you have any questions on that? So then, the question was, okay, can we use Comer to do SNP evaluation, which is something that people are looking at. So, it says, okay, I have these SNPs, might be on the target of microRNA. I'm not sure if it's a real target. I'm not sure what effect it might have on the microRNA target. Can we do an evaluation? So, in order to do the proper evaluation, of course, you need to have SNPs from the same patient than you have the expression data, and we don't. As a second best option, what we did, we said, okay, let's do a focused research. We have some collaborators interested on the estrogen receptor pathway. So we said, okay, let's look at the genes of the estrogen receptor pathway. We don't have SNPs from patients, so let's go to dbSNP and see what SNPs we can find there. And if we can predict any microRNA to be affected severely by a SNP based on the Comer scoring. So we rank the SNPs we took. There are 9 estrogen receptor pathway genes. 15 SNPs on these 9 genes have minimum allele frequency more than 10%, which is a kind of what most people consider as non-rare allele. So for all the microRNAs, if you look on every microRNA individually, you get 29,000 pairs of microRNAs with these 15 SNPs, the targets on these 15 SNPs. And then we applied another filter here. We ended up, we reduced this number, we ended up with 52 SNPs. Now, the way that we ranked that here was this formula, which is the probability that Comer gives, the SVM probability, for the wild type times the difference on the 2 probabilities. So here, practically, we want the ones to come on the top when the probability of binding or regulating is very high on the wild type, And also, the difference between the two probabilities, wild type and SNP, is also very high. These will be the SNPs that will come on the top, okay, of the scoring. That's one method to do. There are other methods to do it. So we found out that one of the SNPs, actually, that came on the top, the one that came on the top, was affecting the seed region of the SRC1, it's also known as NCOA1. And this was a target of miR188 5' or star at the time. Now, the interesting part with this SNP is that it was not included on SNP chips on studies that they had done before, but other SNPs in the area were found to be in linkage disequilibrium with osteoporosis, with bone density in postmenopausal women. So we thought that, that's interesting. So we did the actual validation of the SNP target. So we have the luciferase expression vector. We added the SRC1 3' UTR with the SNP and without the SNP. And and we showed that in the cells there is a difference in protein production on that by itself. And then when we transfect with miR4885P, then the difference in expression of the SNP and the wild type becomes significant. So this kind of proven the target. That's the way to actually prove the target on that. Although in this case, of course, you have proven the target, you don't know the concentration of the microRNA because you put a lot of microRNA in the target. Then they looked on a different cohort of patients, and they found actually that this SNP also, where is the SNP 3708, where is that? 1777. This SNP also comes up as significant on an independent cohort of the study. So the conclusion from that is that, of the whole thing, is that current microRNA target prediction algorithms have two deficiencies that I mentioned before, and solving them can lead to improved prediction and better overlap between the algorithms, and also that the convinced scoring scheme can efficiently rank regulatory SNPs affecting microRNA binding. So, and we recently put up a server, it's still on the beta version, but you can see it from www.benoslab.pip.edu/Cobier. You can find it, you can upload, it's still beta, so sometimes might run slowly. You can upload either one set of SNPs with one expression value, so in one condition, or in multiple conditions. The one before the last part of the talk is about networks. Okay. Generally, when we try to infer networks from gene expression data, people follow one of the two directions. One is what's called relevance networks, which is built connections by looking on pairwise correlations. So you look two genes going up and down at the same time, you assume, if they are statistically significantly up and down at the same time, you believe that there is a connection between the two. Now, so up or down, and in this case will be a positive connection. If it's one going up when the other goes down, then you have a negative correlation, negative connection. Now, the problem with that is there are multiple problems with that approach. One is that you miss the directionality, so you don't know if A regulates B or B regulates A. The second is you usually get high false positive rate. The third one is you miss the higher order interactions, and by that mean that when you see two things going up and down at the same time, A may regulate B, B may regulate A, or another variable, C, might regulate both A and B. You will also see A and B going up and down. So now to solve this problem, smart people do Bayesian networks. I'm not one of them. Smart people do Bayesian networks that they try to sort out by doing a set of conditional independence tests. They sort out these kind of higher order interactions. Now, the problem with this approach, of course, is that it requires large data sets. Sometimes they are not available, sometimes they are. And also you need to have some kind of heuristic apply some kind of heuristics to find the structure of the network, even in these cases, because searching on all possible networks, it's an NP-complete problem, if I'm not completely mistaken. Right. So what we did is, okay, we say, let's do the easy thing, let's do a relevance network, but let's try to cut down on the false positive rate. And how do we do that? We do that by including a pre-calculated prior network information on we combine, let me put it that way. We find the dynamic network interactions, positive or negatives, between pairwise comparisons of nodes, but then we overlay that with some p-value calculation of how much I believe that this transcription factor regulates this gene or this microRNA regulates this gene overall. Okay. We are talking about regulatory networks only, so we are safe there. So in other words, on that part, we provide the directionality and some prior strength, usually not very accurate. And that part down provides the posterior strength, that's more accurate in terms of combination. Now, the static network, we calculated with a bunch of different methods. One is to just bioinformatics approach, to look on whether transcription factors have binding sites by scanning the promoters of the genes or the microRNAs. We look at the literature. So this is just a potential score for regulatory interactions. And in the current version, that was before COMIR was developed, we do a simple voting scheme for whether a microRNA regulates a gene or not. As I said, the posterior information, the dynamic part of the network, comes from the data themselves, from pairwise comparisons. And we also have put up a web server that you can use called miRNA. You can upload currently, I think it supports human, mouse, and Drosophila, I believe. You can upload the mRNA set, the microRNA data set. Here is the prior weight. As I said, we don't have much trust on the prior information, so we put 0.3 as a weighting factor for the prior network, but you can play a little bit with that, if you like. And then, of course, you get these nice and, as I said, completely useless hairballs. But then you can search, you can use that not for knowledge discovery, but at least for hypothesis generation. If you see something that you know and it's interesting, you might like to follow it up. It also calculates a bunch of feed-forward loops, because we know that these are important motifs in network biology, and I will show you an example in a minute. So that's pretty much the summary for that. Yes, sir?
Can you please repeat how you computed the directionality of edges?
Well, the directionality comes from the fact that a transcription factor or a microRNA are the only ones that they are allowed to regulate. So these are the causes.
So it's microRNA to gene, but
microRNA to gene or to transcription factors, and transcription factors to other genes or to microRNAs as well. So here is a transcription factor in the promoter of a set of microRNAs.
But when you have two microRNAs and they have an edge, how you determine which one?
No, you cannot have microRNA to microRNA. There is no microRNA to microRNA edge. No. These are cut out by the algorithm because they have a prior information of 0. Okay? Any other questions? Okay. So, and the last part is, okay, how these are relevant to biomarker discovery and patient treatment. I showed you already something about biomarker discovery, and actually a more causal biomarker discovery when we saw the COMIR and the SNP discovery, right? So we found the SNP that was not only a biomarker, but it also we found a possible mechanism for action of the SNP. So it's what I would call a causal SNP for the disease. Now, I'm going to show you now another example of mechanism discovery. So why do we do all of this? As I said, at this point I have shown you a bunch of algorithms that we have developed over the last few years that are related with microRNAs and so on. So we always have in mind people like those. These are people suffering from a disease called pulmonary fibrosis. They have all given informed consent. And these are all in the cohort we have in Pittsburgh through my collaborator, Naftali Kaminsky. So, and this is also published, this is our success story, it was published in the American Journal for Respiratory and Critical Care Medicine. Now, idiopathic pulmonary fibrosis is a chronic progressive lung disease The median survival rate without transplant is 2 to 5 years. Uncertain etiology, we don't know exactly what is. There might be a genetic background, but we're not sure what this might be. And it's characterized by formation of fibroblastic foci and accumulation of extracellular matrix and irreversible destruction of the lung parenchyma. Wind signaling and TGF-beta signaling is supposed to be involved in the disease, the facilitation of this disease to some point. And another closely related process is what's called the epithelial to mesenchymal transition, where again you get TGF-beta being a regulator of this process. And in this case, you change the fate of the epithelial cells. The fate of the epithelial cells change in become mesenchymal. So they lose their attachment markers. They can become mobile. They can proliferate. As I said, TGF-beta is one of the inducers of this epithelial to mesenchymal transition. So this is the TGF-beta pathway. Please remember, I'm going to ask you these names at the end of the talk, so let's see who will remember the most. Or to simplify the thing, let me tell you what was known at that point when we started working on this kind of network biology. So we know TGF-beta upregulates SMAD3, SMAD4, which upregulates HMGA2. Normally this will downregulate ID2, which will downregulate TGF-beta, and TGF-beta will upregulate ID2. Now, if the concentrations are right there, everything was working nicely and everything is in balance. No harm with that. If you get a lot of these guys, though, then you go to epithelial to mesenchymal transition. So we wanted to see if there is any loop that involves microRNA genes, any feed-forward loop that involves microRNA genes that might interfere with this kind of pathway in some way. Now, we are interested on these particular forms of feed-forward loops because these are the two type of loops that they enhance the primary signal. So here you get upregulation of this gene directly and indirectly by downregulating its downregulator. And here you get the opposite effect. You get downregulation of this gene while at the same time you upregulate its downregulator. So in both cases, when you get a lot of these guys, then you change forever the fate of the cell or the network that was there. Okay. So we were looking for these particular feed-forward loops. So by applying similar ways like in miRNA, before miRNA was there, actually miRNA was based on the experience that we have through that, we found LED7D that is actually, we finish that one, that is actually, we predicted to target HMGA2, and also we predicted that SMAD3, SMAD4 might regulate LED7D. So this is one of the types of networks that we were looking for. Now, look, if you increase the TGF-beta concentration, then this will increase. If this increases, this downregulation will be strong. If this downregulation is strong, this will go away. If this goes away, this will go up. The reason that this goes up is that there are 5 or 6, I think, binding sites of LED7 on the 3' UTR of HMG2. But regardless, this will go up. If this goes up, this will increase the downregulation of the ID2, right? Then once this goes down, this will go down. So then this will increase. So then you get into a loop that you cannot get out of, right? So, and we know that when this happens, then a cascade of other things happening, and at the end you get EMT. So we wanted to see whether this is true. So our friend and collaborator Naftali Kaminsky, in his lab, he did the following experiment. He said, okay, I will knock down the LED7D and see whether the cells will go to epithelial to mesenchymal transition. And they did. These are the mesenchymal markers that they are upregulated when you get just blocking LED7D, no TGF-beta involved. Okay. Okay, and this is how the protein images of this picture. Also, he found that LED7D is one of the downregulated genes in patients with IPF. So the next question was, okay, can I do that in mice? If I block LED7D This is what he did. Block LED17 in the trachea of mice, and 4 days after, did it repeatedly, 4 days after, the mice lungs become fibrotic, and confirmed that with the increase of collagen marker and HMG2 in the lungs, as well as with the tissue examination. So what we saw is we starting from a cell system, we find some interesting feed-forward loop involving a microRNA gene. We saw that this is true. I omitted some experiments that he did to confirm that our finding was real. Then blocking LED17 in the cells and show that they become mesenchymal, and then blocking the same microRNA in the trachea of mice and inducing the pulmonary fibrosis phenotype. So if you block LED17, you induce pulmonary fibrosis. Can we reverse the process? Can you give to the people the LED17? And this is where you go to the development from markers and mechanisms to develop drugs. And the answer to that is maybe. We don't know. We don't know yet. There are experiments going on, but Nathalie and I, we filed for a patent for this gene to be used in pulmonary fibrosis. There is no other cure so far. There might be other genes that are also important. But yeah, so that's the whole story. So the summary of this part of the talk is, as I showed you before, the Comey scoring can be used to evaluate SNPs, and we found that this particular SNP affects the 488-5P association with NCOA1 and is likely to be related to osteoporosis. And as I showed you now, microRNAs are key players in regulatory networks in the lung. TGF-beta controls the expression of LED17. I think that time was the first time that we saw that the growth factor directly regulates a microRNA gene. And LED7D downregulation is sufficient to induce epithelial mesenchymal transition in vitro and IPF-like symptoms in vivo, in mice. So the main conclusions from the talk is microRNAs are important regulators in many diseases. I think we established that. Probabilistic modeling of microRNA stem loops is efficient across taxa. From human trained model, you can predict even plant microRNA genes. Quantitative modeling of microRNA targeting improves the prediction efficiency and the overlap of the existing algorithms. Relevance networks with priors can be efficient tools for hypothesis generation. And COMIR can be used to rank SNPs affecting microRNAs in disease. So with that, I'd like to acknowledge my collaborators that I had the privilege to work with the previous years. As I said, Sabah Kadri was a student in the joint CMU PIT program in computational biology. Now she's a postdoc in Broad Institute. And the CMIR study was done in collaboration with Veronica Hinman, who works on modeling regulatory networks in sea urchin. The COMIR and the osteoporosis SNP evaluation was done with Claudia Coronello, who was a postdoc in my group, and she's now at the RIMED in Italy, in the Research Medical Institute in Italy. And this study was done in collaboration with Natalie Kaminsky, Steffi Esterreich, and Gary Stormo. Gary Stormo is my former postdoc advisor. The MIRConnect was done by Grace Kwan and Hari Afanasiou. Hari Afanasiou wrote the server, Grace Kwan developed the algorithm. And the IPF study was done by David Corcoran, who, in collaboration with Natalie Kaminsky, who David is now the director of the core of the core facilities, data analysis core facilities in Duke University. These are some of my previous trainees. This is Sabah Kadri, and this is David Corcoran, whose work I presented to you. So if you need to send me email after we leave, this is my email address. This is the email address of the lab. And with that, I'd like to thank you very much and take any questions. Who will go first? You, sir. You understood everything? Was it that clear?
So I have some difficulty with the notion of relevance networks being used to induce regulatory networks.
Mm-hmm.
What the relevance network representation is just a visualization of associations that are univariate associations. And as such, for every true causal relationship that you include, any direct causal link, you get also all the indirect ones, you get all the passengers,
Mm-hmm.
you get all the downstream effects,
Right.
you get all the confounded ones.
Mm-hmm. But when
it's
you use prior, you cut down on most of those.
correct, but when you start with something that's so fundamentally flawed, then the use of a prior becomes a very difficult exercise. Your criticism, if I understand this correctly, is that let's not use proper causal induction algorithms, causal graph induction, because it's worst case intractable. But
No, the worst case is that we don't have enough data. So sometimes you look on 25,000 variables, and you look for 10 samples, 50 samples, the maximum. And sometimes, even though I think this is not, you can use perhaps Bayesian network using the prior information. That might be much better. So those are not the relevance networks with prior information. So you can use the prior information and incorporate it in the Bayesian network. That might be an interesting thing to do.
how much prior information you must have in order to decipher the induction of a network that, I mean, the hypothesis space is near infinite. And how much prior information is needed in order to make this judgment call. Whatever applies for the general problem of induction of a causal graph, for any algorithm, applies for any other methodology, be that may including priors or not priors or data-driven. I mean,
right. So, but if you don't have that, for example, associations between microRNA and microRNAs might be there. Associations between enzyme and a microRNA or enzyme and a gene might be there. Which we know that in a regulatory network this cannot happen. At least they cannot be direct interactions. Could be a kinase that phosphorylates a transcription factor, which then regulates something else. But the kinase itself cannot regulate something else directly, right? Because the function of kinase is not transcriptional. Right? So that's why I say that with all these networks, if you apply them blindly and you don't have any prior knowledge at all, the prior knowledge can be also 0-1, right? What are the allowed connections, what are not allowed? But I would think that if you have some p-value calculation already, not 0-1, but something between 0 and 1, and you can use that as prior in a full Bayesian network, and that will give better results. That's my prediction.
okay.
Right. Otherwise, you get strange results. If you're interested for regulatory network, yeah. If you're interested for any kind of network, any kind of interactions, that's a different story.
There is a literature on the ability of a variety of properly designed and not properly designed algorithms to induce regulatory networks as well as broader causal networks. They give a sense of how possible this is under assumptions about the density of the network, the distributional assumptions, the linearity or not of the relationships, and the sample size that you have. I think will be tremendously interesting over time for people who are more stronger advocates of, say, using prior information, just like the experiments in this talk, to give a similar sense of like broad experiments that can show how these techniques work.
Right. Yeah.
Because then we would put them on a on a side-by-side comparison and
Yeah.
make sense out of them.
Yeah, I agree. Yes, sir.
Is it possible to use, it's a question about miRNA. Is it possible to perhaps a sequence of miRNA? Can I use miRNA to know which target, potential target, which one is the best target, or
So
it's a viral miRNA.
Right. So you can download the data. They are graphically displayed as I saw it there, and you can zoom in, out. You can select part of the node, part of the network to see what genes are. But also you can download them as a tab delimited file.
It's because all of the miRNAs that we know
Right.
are in this database.
Right. Yeah.
It's an HSV1, it's an herpes database.
Right. So it contains all the known microRNAs, but then your expression values on the microRNAs will determine which microRNAs will be included on the network display. Right? Because you submit a file of mRNA expression and microRNA expression. This might or might not be complete. You might say, I'm interested only in those mRNAs and only in those microRNAs. Can you find the connections between them? Right? If you submit the full list of mRNAs and microRNAs, then yes, all the known ones will be there. Everything that you submit, the algorithm does not have any prior knowledge of what you submit. We'll take it as label and will plot what is it to plot. The only thing that it needs to be with a standard name is because of the prior information, right? So it needs to match what it knows as prior information with what you submit as expression.
So I can put a sequence that it's supposed to be a microRNA.
Not a sequence, no. You need to have a name, a label. Right? Now, if you want to use the prior, because you can run it by putting the prior weight to 0, so it doesn't need to run with prior. Right? If you don't want to use the prior, then you can put whatever labels you like. But if you want to use the prior, then you need to have the names that they are the standard names, so they can be displayed.
So, do you think that, like, by using algorithms, can we using an algorithm and saying that there are no off targets? Is that sufficient?
There are no what?
Off targets. Is that sufficient, or because sometimes, like, reviewers ask for, like, the biological confirmation of this, even though, like, an algorithm or 2
Right.
or 3 say no.
No, the algorithm, especially when it goes to microRNA targeting, as we saw, they do make a lot of mistakes. They have a lot of false positives, right? Even target scan, it's very sensitive, it's not very specific.
So even though, like, 3 different algorithms tells you that there are no off targets, you should do still the
So this is what most of the people doing, is that they combine multiple algorithms, hoping that the set will become smaller and smaller, but it will become also more biologically correct, if you like. I think I remember I read a study that shows that this is not exactly true. And I don't remember now exactly. Okay, thank you very much. Thank you very much.